Okay, today we will be talking about origin of life. Through time immemorial, people have wondered on how life originated. That is, how life began on Earth and how they developed. So, in this context, we will see what is life cycle, and then we will understand about what is abiogenesis or origin of life, that is chemogeny and biogeny. And we will learn about the steps of abiogenesis. And then we will understand what is hot dilute soup theory. Then we will understand what are protocells or probionts. They are the coacervates and microspheres. Then we will understand what is evolution, what is variation, what is the difference between variation and mutation. This is today's target. So, what is life? Life can be defined as a distinctive characteristic of a living organism from dead organism or non-living thing, which can specifically be distinguished by the capacity to grow, to metabolize, to respond to stimuli, to adapt, and to reproduce. So, living organisms grow, they metabolize, they respond to stimulus, they adapt to their surroundings, and they reproduce in numbers. And this leads to the term known as life cycle. Life cycle is the series of changes that the individuals of a species undergo as they pass from the beginning of a given developmental stage, generally which is an embryo, to the inception of that same developmental stage in a subsequent generation. So that is all the stages in the whole life of that organism, from birth to death. Instead of death, from birth to new birth. That is also life cycle. So of a life cycle of a human, they include pre-birth, in your in the mother's womb, birth, early childhood, middle childhood, late childhood, adolescence, early adulthood, midlife, mature adulthood, late adulthood, and death. In between, there is reproduction. This is the life cycle of a human. Now, let us understand what is abiogenesis or origin of life. In biology, abiogenesis or the origin of life is the natural process by which life has developed from non-living matter, such as simple organic compounds. That is, from non-living organic compounds, how they assembled, and you have the phenomenon of what you call today as life. Abiogenesis consists of two phases. It has two phases. The first one is the chemical evolution, or it's known as the chemogeny. In this particular chemogeny, chemical evolution, there was formation of complex organic molecules from simple organic molecules through series of chemical reactions in the oceans during the early stages of the earth. This was the first step in the development. And the next step is the organic evolution, or it's known as biogeny. It is the formation of protocell, the first cell, and subsequent towards the modern developments towards the modern eukaryotic cell, from protocell to modern cell. That was organic evolution or biogeny. First is from complex organic molecules, they assemble to form the first cell, first stages of life. The organic complex molecules were formed from simple inorganic molecules through chemical reactions in the oceans, and then biogeny, where the first cell was formed. So what are the steps for the origin of life? The first one was chemogeny. So first, there was presence of free atom in the environment. Free atoms were present in the environment like hydrogen, carbon, nitrogen, etc. There was formation of simple inorganic molecules. Simple inorganic molecules were formed from the free atoms like water, NH3, etc., ammonia, etc. And all these events happened in the prebiotic oceans. It formed a hot dilute soup. Now, from these simple molecules, inorganic molecules, there was formation of complex monomer organic molecules. Then complex organic molecules, monomers, were formed like simple sugars, amino acids, etc. And then after they were formed, the complex organic polymers were formed. From the monomers, complex polymers of organic molecules were formed, like polysaccharides from simple sugars, proteins from amino acids, etc. Now, the steps of origin of life. We will talk about biogeny. Biogeny included the formation of the first protocells, where complex organic polymers they aggregated together to form the earliest form of cells. That is, they form the coacervates of the microspheres. The next step was the evolution of protocells to prokaryotic cells. Protocells were upgraded to form prokaryotic cells, having no true nucleus and any membrane-bound organelles. That is, they were the earliest form of living cells. Then there was the formation of eukaryotic cells. With the help of evolution, prokaryotic cells evolved to advanced eukaryotic cells. And finally, it gave rise to multicellular organisms. From unicellular cells, advanced multicellular organisms originated. Now, what was this hot dilute soup? The theory of hot dilute soup or primordial soup, which was proposed by Haldane to describe the prebiotic, that is, before the life formation, to describe the prebiotic oceans, where organic biomolecules were formed from simple inorganic compounds by the combination of carbon dioxide, ammonia, and UV radiation. Next, Oparin and Haldane, JBS Haldane, thought that with the mix of gases in the atmosphere and the energy from lightning strikes, amino acid could spontaneously form in the oceans. This idea is now known as the primordial soup or hot dilute soup. The next thing which we will say you have to study is about protocells or probionts. Now, what are these? Protocells or probionts, they are self-organized, endogenously ordered, that is all ordering comes from inside, spherical collection of lipids. Researchers assume that modern living cells were initially originated from protocells. So, according to Oparin-Haldane theory, simple molecules they polymerized into complex molecules, and then these molecules formed aggregates which were known as coacervates or microspheres. There are two theories behind protocells. So, coacervates and microspheres are cell-like structures and they resemble living cells, but they do not show all the properties of cells. They form spontaneously in certain liquids and they are surrounded by a membrane. নিজে থেকেই spontaneously তারা তৈরি হয়েছে. Now we must understand what is coacervate. Coacervates are membrane-bound vesicles like microscopic structures. They are formed by the aggregation of complex organic compounds, mainly lipid compounds. They resemble living cells. However, they do not contain hereditary materials like DNA and RNA. Coacervates, surrounded by a membrane like a boundary, they are able to take substances from their surroundings and grow in size. Once grow into a certain limit, they divide and form new coacervates. थोड़ा इतना बड़ा होके एक लिमिट आया and then it will divide and form new coacervates and बाहर से मटेरियल लेके और बड़ा होगा. The word coacervate was introduced by Oparin, and according to him, coacervate is a structure which consists of a collection of organic molecules which is surrounded by a film of water molecules. He said coacervates are a kind of protocells. Next, we come to the theory of microsphere. Microspheres are tiny droplet-like structures made from the aggregation of organic molecules, especially proteinoids or amino acid molecules. The term microsphere was introduced by Sidney Fox. According to him, the microsphere is a non-living collection of organic macromolecules with double layered outer boundary, and this layer is non-lipid. This is a figure of coacervate and microsphere. On the left, you can see this is the coacervates. You find small coacervates in specific solutions. And on the right, you see microspheres, which you can create them experimentally under certain conditions. They can be created, but these structures were thought to be initial protocells. We can recreate them now in a laboratory, but it is assumed that from the hot dilute soup, these structures started to form. So the key differences between coacervates and microspheres is that the coacervates have a single membrane while the microspheres have got double membrane. Furthermore, the coacervates are aggregates of lipids while the microspheres are aggregates of proteins. एक प्रोटीन है, एक लिपिड है. You know the difference between prokaryote and eukaryote cells. So I am not going into details on this. And now we come to the question, what is evolution? Biological evolution is the change in the heritable characteristic of a population over succeeding generation, and which relies on the process of natural selection. 60 million years in the evolution of horse, you had Hyracotherium. 40 million years ago, you had in the evolution of horse, Mesohippus. In 30 million years, you had Merychippus. 10 million years ago, you had Pliohippus. And the modern horse today is the Icus. You can see very clearly the characteristics which have changed in the leg of the horse over the span of 60 million years. And what is variation? Variation refers to differences or deviations from the recognized norm or standard. It can be a modification in structure, form or function of an organism, deviating from other organisms in the same species or in the same group. थोड़ा सा अलग है. In genetics, variation refers to an individual that possesses characteristics different from the others of the same kind. Genetic variation usually arises as a mutation in a gene that encodes a protein or an RNA. For example, a person's skin color, hair color, dimples, freckles, blood type are all examples of genetic variations that can occur in a human population. So differences between variation and mutation. So mutation is the natural permanent change causing changes in the DNA or genetic sequence in any living organisms. These changes can be small to large, which may affect the entire genes or chromosomes. On the other hand, variation or genetic variation is seen in groups or population. In a whole population में अचानक देखा जाएगा एक particular change आ गया in genes as well as in alleles. It can be due to environmental factors and intensify the process of natural selection. Variations bring evolutionary change. So do you people have got any questions? कोई question है?
No, sir.
No, sir.
नहीं सर. नो
Okay.
सर.
Okay. Okay.